Thank you, Kelsey, for that extremely generous introduction, and I really appreciate the opportunity to come here and talk today. I think I've been to every Autism Science Foundation Day of Learning, so it's great to be here. So as we've heard, and I think as everybody knows, autism spectrum disorder, as it's currently defined, is extremely broad. So you might have an individual who's highly independent and verbal, and whose autistic features might help them succeed professionally despite being socially quirky. On the other hand, you might have an individual who's nonverbal and has very high support needs. And the communities represented by such individuals are not always on the same page on things like research priorities, and I think that's actually a vast understatement often. And it's confusing when each is advocating for its own specific needs, but what's heard is that their needs reflect all of autism. So what are the challenges here? Well, for researchers, treating autism as a single group means they're forced to average biology across what are likely very major differences, and this dilutes signal, and it makes it extremely challenging to perform any patient cohort studies where people with autism are treated as a single entity. And one metaphor that comes to mind that I think maybe helps to think about this is sort of a bowl of mixed fruit. And if one were to ask, well, what's the average flavor or texture, it really doesn't make any sense. And for families, this heterogeneity is also problematic because it means that there's a lack of information about predictive trajectory and the effectiveness of potential interventions. And I think that's really what families want. So I think maybe the height at which this problem has come to these days is epitomized by this recent quote from Dame Uta Frith, who's the emeritus professor at University College London and played a large role in actually defining the autism spectrum, where she said that autism study is her life's work, but the spectrum has lost all meaning. And this was just in the Sunday Times a few weeks ago. So why is this lumping splitting problem so complicated for autism? Well, let's compare it to heart disease, for example. Heart disease is defined and diagnosed with biological tests that are objective. So familiar ones are cholesterol and blood pressure screening. So those thresholds may change over time. They changed recently for cholesterol, where the sort of cholesterol targets were recently changed. But the underlying tests themselves are stable. The problem with autism, or the challenge with autism, is that it's behaviorally defined. It's based on criteria from the Diagnostic and Statistical Manual, the DSM, and that manual has changed many times over the last 20 years or so, often for good reasons or for well-intentioned reasons. And I think importantly, the types of tests that are used are observational, so they're done by a clinician or a caregiver, which means that they have a necessary subjective component. They're often done in clinical settings and time-consuming. They're episodic. They're done from time to time, so a kid may be seen once a year or every couple years. And critically, they're distant from the core biology, and I sort of make the comparison to heart disease, where we're thinking about blood pressure and cholesterol, and we understand to some degree the mechanistic role. But the kinds of tests we're talking about in autism are more distant. Critically, also, autism unfolds over the course of development, so we're dealing with a moving target. And I think possibly most crucially, and potentially has gotten less attention, behavior is really an emergent property. So it emerges from an interaction of the developing brain with the environment, and those are highly interconnected relationships with parents, siblings, peers, and society at large. So there's some exciting news on the horizon. There have been a couple recent papers, actually about 5 or 6 in the last 2 years, that have attempted to divide the spectrum into potentially more rigorously defined and meaningful biological categories. And these are based, what's the magic sauce here? These are based on large cohort sizes, the use of genetics, and critically new analysis methods that include things like machine learning and AI. And I've listed 2 papers that I'm going to come back to in a moment that I think epitomize some of the progress that's being made. So why should we care about different subtypes? I actually think we just started great in the previous discussion, we heard a great reason why, but the mantra of precision medicine is find the right treatment, and that could mean a behavioral intervention or supports or medicines for the right person at the right time. And to get anything close to this for autism, we need to divide the spectrum into meaningful categories. So going back to the fruit bowl analogy, we need to know whether a kid is a raspberry or a pineapple or a blueberry in order to think more constructively about this. So I'm going to start with a small detour to talk about genetics. Because for the last 20 years or so, there's been a tremendous progress in our ability to study the genome from just a few drops of blood or cells in saliva. And this has evolved from relatively biased approaches where researchers had to choose sort of candidate genes and then ask whether that gene was involved, to largely unbiased approaches where we can now look at the entire genome. And the cost for doing so has plummeted at the same time, allowing us to do this at scale. All of those things are crucial. And importantly, genetics help set the stage for who we are. So think of identical twins. Think how similar they are, both in what they look like and how they behave. And in fact, twin studies in autism have motivated much of the modern genetic effort. And just because of some previous comments, I want to highlight that, for example, identical twins, if one twin has autism, the chance that the other identical twin has autism is about 80%, but the rate is very much lower for fraternal twins. And that's the crucial difference in terms of telling us about the role of genetics. Okay, so I'm going to try to summarize 20 years of genetics in 1 or 2 slides. And one of the highlights is that about 10 to 20% of individuals that have a current DSM diagnosis of autism carry a genetic change that isn't present in either biological parent, and that's referred to as de novo or new. There are a few hundred locations in the genome where DNA changes confer a higher likelihood of a diagnosis of autism or related neurodevelopmental disorders. I want to make 2 critical points in the context of subtyping. There are many genes, but they're potentially distinct biological processes that are more limited that are represented by these genes. And then critically, the magnitude of the risk that these de novo genes that I'm talking about confer is very significant, on the range of about 50 times increased chance that someone who carries one of these changes will go on to develop autism or a related neurodevelopmental disorder. One of my colleagues likes to call this genetics with a capital G. So why am I belaboring this point? Well, in the context of subtyping, for the 10 to 20% of individuals with autism who have one of these genetics with a capital G changes, that may be the most relevant category for that individual. And in fact, many patient advocacy groups around these particular genetic changes have sprung up over the last 10 years, and with associated research studies. These have names like SCN2A or SHANK3. I actually saw some information in the back about this. And the Simons Foundation actually has a project that tries to be an umbrella project for many of these called Simons Searchlight. And critically, there are therapeutic possibilities on the horizon that are gene-specific. And these further support that these gene groups are meaningful. And in the next talk, I think you're going to hear much more about this. So this is an interesting setup for that. But for many or most on the autism spectrum, they won't have a large genetic event. And one way of thinking about this is imagine a glass of water with different size ice cubes, and if the water overflows or passes a particular threshold, a person would have a diagnosis of autism. So the large de novo genetic events that I've been talking about, like SCN2A or SHANK, are equivalent to one of these large ice cubes. Almost on their own, often with some other small effects, they can sort of push someone past a threshold for diagnosis. But for many other people, the risk factors are comprised of smaller genetic events, each of which has a much smaller effect, but it's the combination of these events with other possible events, like environmental events and other things that are harder to study, that will push someone over the edge. And this total risk from these small genes is referred to as a polygenic risk score, and I'm going to come back to this in the next couple slides. So what's really exciting is that there have been several new approaches that people have taken to try to use these new methods to subtype people. And I'm going to first talk about a paper from Littman, Sauerworld, and Troyanskoya, and I want to say up front that the authors, some of the authors have an affiliation with the Simons Foundation, so just full disclosure. This was published in Nature Genetics last year, and it took what they refer to as a person-centered approach. And instead of starting with genetics, they started with behavior and took a cohort of about 5,000 autistic children and looked at 239 measurements that were made of these children. And these include things that everyone with family members with autism would be familiar with, that include things like age of walking and various pieces from different diagnostic tests. Then they used a statistical approach called mixture modeling, and they came out with, from this data-driven approach, with 4 different groups based on this behavior characterization. And they referred to these as, in the yellow here, with moderate challenges, those who are broadly affected, those with social and behavioral mixed, and those with mixed autism and developmental disorders. So many people have tried using techniques like this to subdivide the spectrum. So what's special about this paper? Well, what was interesting right off the bat was that this data-driven approach that they used mapped onto categories that made sense clinically, essentially. So for example, this red group, which they referred to as broadly affected, had high rates of developmental delay, self-injury, anxiety, and mood symptoms, for example, broadly affected across the board. And for example, the green group, which they referred to as social behavioral, had a different profile. So these profiles sort of make sense from a clinical perspective. But critically, they also found that the polygenic risk profile for each of these different groups differed from each other. So that's important because that tells us that the genetic makeup of these different groups might be distinct. And they matched clinical profiles where you can calculate polygenic risk scores for other disorders like depression, for example. So what's so exciting about this is it gives us some clues to the biology. For example, the cell types that might be involved, and this is in humans, answering questions like where and when. And importantly, this result was replicated in another cohort, which is critical. The other paper that I just wanted to mention briefly from Varun Warrior and colleagues that was published in Nature last year took a different approach where they looked in a variety of cohorts and divided individuals, those who are early diagnosed and late diagnosed. And of course, the diagnosed is really just a proxy for the emergence of symptoms, or a hopeful proxy of that. And they also found that the polygenic signal for these different groups, the early versus late group, differed, which suggests that the early versus late essentially have a different genetic makeup, which is really interesting. And it also mapped onto these differences in mental health challenges. So for example, the late diagnosed had an increase of major depressive disorder and a corresponding higher polygenic risk score for that. There's another whole way of looking at subtyping that's common in medicine, and I think also might be something to keep an eye on in the autism and neurodevelopmental field, and that's treatment response itself being a subtype. And I'm just highlighting one paper here that came out just earlier this year, which basically looked at a differing response to a drug called bumetanide. I'm not endorsing this, I'm just using it as an example of a different mechanism of potentially subtyping by identifying responders and non-responders in treatment cohorts. And I think this is something that we have to incorporate moving forward. So what might the future look like? Well, I want to emphasize that I think the whole lumping, splitting problem is going to remain very dynamic over the coming years. This is, I think, far from settled science. Maybe in the future, instead of autism, there'll be new descriptions that are more meaningful to families and researchers, that are likely based on a complex integration of genetics and more sophisticated behavioral and multimodal biological measurements. And what might those measurements be? Well, I think it's going to be important that they be as objective as possible, as continuous, passive, so measurements, for example, using, I think I have a picture here, using things like smartwatches and cell phones that people carry in their pockets that are measuring various kinds of data, physiological data, for example. They'll be scalable and longitudinal, so scalable so we can do this in really large cohorts, longitudinal so we can do this over the lifespan, starting young and including adult trajectories. And critically, I think they'll be closer to the core biology, so measuring things like sleep and sensory behavior and motor behavior that might give us more insight into the biological mechanisms. They'll no doubt require some of the advances that are rapidly being made in sort of big data, AI, and dynamical systems approaches. Hopefully they'll be predictive, so measurements made early in a child's trajectory will give some indication of how that child will end up. And I just want to emphasize that I think there'll still be a need for qualitative and patient voice inputs. That's always been really fundamental, and I think that will continue to be. So a couple final thoughts. I think a heavy dose of humility is required here. Biology, and especially the brain and human development, is just beautifully messy and complicated. I included a picture here of just some neurons in the cerebral cortex. We're dealing with something really, really complicated. Real progress takes time. We all have a sense of urgency that we want answers quickly, but biology is complicated. I want to acknowledge also that the healthcare system of the real world is really complicated and slow to adjust and accommodate new measures and classifications, but I think we need to really be all pushing hard on that. And I want to end just with a call to action for research participation for all families with autism and neurodevelopmental disorders. I think ideally every kid who has a developmental difference would be part of research, and I am hopeful that the emergent technologies that I've talked about will lower the barriers to entry. So thank you very much.
Hi, Larry Hillebrand. I'm wondering if you could relate your interest in tools and splitting the population with undertakings to develop autism-related biomarkers. Should we think of that as the same thing, or is what you're advocating a subset of it or a superset?
It's totally related. People use the word biomarker in many different ways, often in the context of a clinical trial setting, so having some objective way of measuring progress, for example, in a trial. But even that, it's a pretty broad word, so there are biomarkers that are related to so-called target engagement, so is a drug getting in or not into the system? Are there changes in behavior in response to a certain treatment, whether it's a drug or not? So it's in the category, but I think I'm using it even more broadly because I think some of these mechanisms, like studying sleep, and other physiological measurements, might give us even deeper insight into some core biology that may themselves not be terrifically useful readouts in a clinical trial, but may give us some insight into different populations of individuals.
Hi, I'm Vanessa Babino. So you spoke about genes and linking genes to environment, but I wanted to sort of bring it back to what I think I understood Jill Hesher speaking to was in terms of environment. So what about like gene-environment interactions that might be linked to these behavioral outcomes? If you've ever, speaking to this body of literature, are they also incorporating? I imagine they are, and your thoughts on that?
Yeah, I mean, there's no question that genes drive very substantial, I think I used the phrase in one of the things, they sort of set the stage, I think, for the biology. And then there's no question that it's an incredibly complex interaction with the environment. And by the environment, we might mean peer or other interactions with family members. We could be incorporating various types of things in the environment, food or toxins or things like that. I think often people have said that the Simons Foundation is overly focused on genes, but I think our view is not that genes explain everything, but they provide a highly rigorous toehold into a system by which you can then study some of these other effects. Does that make sense?
Yeah, I was just curious if there was any, environmental components included in the research, but yes, I understand the response that it is complex. Thank you.
I think there's a question over here.
Thank you very much, and I think that makes a great deal of sense looking at subtyping the spectrum. But my question is, have you given any consideration to how that may affect positively or negatively the levels of services that are provided to the community?
Yeah, no, it's an incredibly important and very fair question. So I think one of the issues in this field that has been really hard to get past is their questions of research. I think we really all want to understand the fundamental biology so that we can get a better sense. That's what a lot of this subtyping is about. But we're not operating in a vacuum, and in the United States in particular, a particular diagnosis is sometimes necessary to get services that we know are important for everyone. So it's hard for me, I'm not in the business of providing services, but it's hard for me to know exactly how to react like that. I think maybe the optimist view is that we need to continue to advocate for the value of services where they're valuable, whether or not someone has a diagnosis of X, Y, or Z. But it's an incredibly fair point and very important. Okay, thank you very much.